me and Dr. Li, we went to Orlando, Florida, from April 8th to April 10th to participate to see the the Fertilizer Show 2025. And there's a lot of people, a lot of companies, and all kinds of equipment, fertilizers, and some academic people too. So we actually met many people. So if you have any question, anything we need to discuss, we can stop anytime. Otherwise, I can share something what we got from that meeting. So they have many fertilizer show or expo internationally for almost every year, every places. This one is in Florida, just next to the Disney World. I don't know if anyone has been there, and we were there like 25 years ago when the kids small. We took them see the Disney World, and after that, I went there several times for the meetings, but I never go into the Disney World again. Well, this time is also the show is next to at the gate of the Disney World, but we didn't go. We just see the fertilizers. So this fertilizer show is nice to meet the people. We know what's the best on the market, what's the best equipment, what's the best technology, what's the best fertilizers, what other people do. So then you see a lot of people. I don't remember how many companies there. Several universities there. University people just for the academic and to discuss all the science. So we went there, and this is one. Well, this is another thing for the fertilizer show for the stuff. So this is the location is Orange County Conventional Center. Orange County is one of the county of the Florida state. It's here in New York, we're so cold, but Florida is so warm. The sky was in New York City, the sky is not that blue. In Florida, because they are next to the ocean, the air is much better. Air quality is very good, and they green, evergreen all the time, like in Africa. In New York, we come back to New York, it's all the trees can't just come back, no leaves. So this is the conventional center. We took a picture in front of the, I don't know, it's a picture. I think it's a picture. But this is the conventional center, part of the conventional. Conventional center is huge. The work inside take forever from one end to another end. And the fertilizer shows on the west coast. So this is a front, but the west coast is on something, another building on the left side. So this is inside. It's like an airport. New York airport is, we come back to New York. New York airport is take a train inside and walk for a long time to get to the other end. Of course, the New York airport, JFK airport, is not the largest. The largest airport is the Beijing airport. Second largest airport is the Atlanta airport in Georgia, in the middle southern U.S. So I worked there for 5 weeks, like 10 years ago. It's very hard. So this is a fertilizer show. This is like International Fertilizer, IF. So this is all their some logos. I took a picture over there. And at the show we met because we are all professors. Dr. Li and I, we are kind of academic, not really businessmen. So we're interested in talking to the professors. In the middle, these two, one is Williamson, another is Joe. So right side is Joe and is Williamson. And this all come from University of Florida. University of Florida is huge. They have many, many campuses. The main campus in Gainesville, and they have other branch. Like we had in New Jersey, we have Rutgers University. Rutgers University is the largest university in U.S. We have 3 to 5 campuses on the main campus. The campuses on the main campus. And also we have several different, like, we still can call campus, but it's like an hour or 2 hours away. And also have all kinds of experiment extension stations. Like last year, we went to the blueberry and the cranberry research station. That station is focused on the blueberry and the cranberry. And locally, we also have peach research station. We also have grassland, soda farm, just to produce all the grasses. They have farm station, and I also have other orchids and have vegetable experiment stations. So University of Florida is like Rutgers University. It's a land-grant university. The land-grant university means that university is U.S. land constitution by the Congress, by the whole government, supported by land, which means land is the core of the country. So it's government owned and the government supported. It's land grant. Land grant is always means is the original, the top universities. So this is the guy we talked a lot with him. Is Professor Joe Dupix. He work on the foraging. He like animals feed, animal what grass, what food they eat. They eat all kinds of different grasses, maybe legume, maybe something else. After they eat, they will change the, maybe soil. If you produce crop for the animals, for the cows, the cattle to eat, and then you will need fertilizer. So you can change the fertilizer to promote the feeding. And if you want to call science, ecological wise, or organic methods. So he was promote a lot of use legume family plants to feed livestock and make it sustainable agriculture. This will reduce chemical fertilizer to the soil, and also the animal and the livestock feed on the legume plants, and also they can give back manure to the soil. So soil is a nice circulation of the nutrients from the soil to animal to manure. Is a nice circle. So he did a lot of work, and he got some awards. So In the future, we may do some cooperation research project with them. He has many graduate students, PhDs, masters, and it's not like the government. We don't have students with me anymore. We used to, when I work at university, we have students. Now I'm the government officials. We don't have students anymore, but he has. So if we need to do some huge research, some small project, try to test some new fertilizers, and we can work with them. Also, we discussed a lot for the future of the fertilizer, future of the agriculture. Basically, it's we Greenwood, we standing on the front line. We are the updated knowledge company. We have all in the front line. We are not any less or behind from the university. I'm still research people, so we are at the scientific front line. Basically, what we doing the same thing what the Florida University people, what they are doing. We are doing the same thing. We are promote organic fertilizers, minimize, reduce chemical fertilizers. This is a trend. This is the future. And this one, Willington Pavan, he is also the professor from University of Florida. This guy on the left next to me is him. So he is also this research professor. There are all kinds of ecological modeling more in the future. We know it's in the study area of the AI, artificial intelligence. The AI time is coming. So we have to have not only organic fertilizer, we not only understand all the system, traditional or conventional plant physiology, nutritional science, or all kinds of techniques. We are now need computer science, the database. So he do a lot of modeling for a lot of computer science, put computer science knowledge incorporated with agriculture. Actually, I was teaching called ecological science for agriculture for many years. We use ecological methods, systematic methods, use computer modeling to find out, understand the agriculture system, find out where is the biological windows, where is the biological strengths, which time, which part are something we can attack, we can work on to improve the ecological system or to apply your fertilizer or apply your pesticides. Apply pesticides, apply insecticides, or apply fungicides, the weedicide. They all need science, need modeling. You cannot just go over there. Okay, let's spray. No, that's the old time. My grandfather's generation. They would do whatever they wanted to do, just to say, okay, I need to do some fertilizer. They just do fertilizer. They don't have data support. So now, in the now time, we need to do all this agriculture practice should be based on the data. The data-driven agriculture ecosystem. That's the future. That is based on your knowledge. You totally understand the ecological system, so you don't do wrong things. Like usually have one time, I went to my wife's cousin's house. They told me one of their trees have a lot of diseases, and they spray the fungicide for many years. Every year they spray, and every year the tree is going down. It's dying every year. So doesn't matter how much they spray, they're dying every year. So that evening, I used the light to look at his plants. That plant, the first things they thought it's diseases. That was not diseases. This is scale. The scale insects. The scale insects like diseases. You think most insects are moving. They have 4 wings and have a head, have body, and have 6 legs. The insect. That's what you know. The dragonflies, the butterflies. This is the insects. The cockroaches. You see the cockroaches running very fast. They have 6 legs. Their legs very long. That's why they're running so fast. The dragonfly can fly so high, so fast, because they have very strong wings. So that you think these kind of insects are insects. And the scale insects, they never move. When they scale the mother, they lay eggs. They put a cover, a wax on top of the mother, and the scale insect. This is the scale insects. They hiding a cover underneath. You don't see. And on the top, it looks like a piece of something color. And the female here, they have most parts is maybe 50 times longer than their body size. If the body is 1 millimeter, the most parts, the needle can be 5 centimeter or even longer. So they only lay eggs. The eggs hatch only a week. These eggs of the hatch, the new larvae, they move, and they move to a new place. They stay there the whole life. They never move again. They put the needle sticking in the plant tissue. They move curve around everywhere. They're sucking the juice up. The whole insect, the scale, they stay there, never move. So that's why it looks like the insect looks like a disease, looks like just a piece of paint. So that's why he was spraying the insect fungicides all the time. It never work because it's an animal, it's not a fungi. So then I told him, you totally did it wrong. You shouldn't spray fungicide. You should spray insecticides. Well, if you spray insecticides, you're not going to kill them either. Why? Because they have a wax shield that is like an umbrella, like a raincoat that covered these insects. It's only 1 week or 2 weeks in the early spring, like April or May, when the eggs hatch. There's a new crawlers, new in tiny, less than a pinpoint, less than 1 millimeter size. This newly hatched scale, they can move. They don't have wax cover. This biological window, every year only have 2 weeks biological window. You can spray, can kill them. After 2 weeks, they settle down. They have wax on top. and then you cannot kill them anymore. So that's why we need to understand the whole system, and we need to find out the biological window. So all this need computer science. Of course, you have conventional. In the past, we don't have a computer, we can do it, we can tell. But if we have a computer database, have a software, put it on your cell phone, put your location, okay, there's Kenya, put Kenya in it, and put Nairobi, put a city in it, then I know what time these insects come out. Okay, this predicted is March 15 to March 20. It's only a week. Then you can open your cell phone, the app, and it can tell you this is the time you spray insecticides to kill that species of insects. This is data-driven agriculture. For fertilizer, same thing. Fertilizer is also, you just receive a lot of new plant cells. We made up for some, what's timing, how much we spray the fertilizer. All this timing, we didn't put a March, June, or January dates on the brochure because everywhere is different. So we put a plant development stage. Put the plants just started or started blooming or fruiting or harvesting. So this is because we are not using computer database software. Someday, some way in the future, sometime in the future, we may be able to develop a software, put an app on your phone. You key in your location, key your crop, and I can tell you what time you use your fertilizer. This way you maximize your fertilizer, you don't waste anything. Not all the time you put a fertilizer on the plant that works, no. Sometimes you put a wrong timing, you may don't have fruiting at all, or you totally waste. Or if in the later season or the dry season, when you dry season, not the rainy season, you spray the cassava, the leaves maybe come back. Then you only spray one time and rainy is not coming, the dry. The newly developed, newly emerging, the leaf may die out. So it's totally waste, maybe can kill your plants. So after talking all this, you will see how important the modeling cropping system it is. It's very important. It's very difficult. You at least you need 5 to 10 years data to support, to set up one of the system to your local conditions. It's a future. Sometimes we may be able to see it or we can use it. Actually, if you work with the universities, academics, I did the first of my modeling in 1986, 88. How many years ago? 40 years ago. That's my first modeling for the farmers to use. So the method is there. We just need every location, every country, every crop, all different. You cannot use one method applied to all. For the development model, you can use it because the crop, the plants, they develop by degree days. The degree days, so 2 terms together. So how many degrees like plants not develop if the degree temperature below certain degrees. So you have to maybe temperature above 10 or 15, the plants can continue to develop. We only use that degrees we accumulate and time days, how many days they got. And it's called degree day model to calculate each crop development stage at each location, each city, each country. So this is all possible, but each crop maybe need at least one PhD student to finish. So that's modeling. We met another professor, Shinsuke. I thought he was Chinese, but in the end, he's konnichiwa. He was a Japanese professor. So he is a professor at the University of Florida. And he's doing more close to us. We are doing a lot more. The Professor Joe, he was doing the animal feeding plus soil. This one, Shinsuke, he was doing vegetables and fruits. So a lot more close to us. But we do a lot of agriculture crops, do the nutritional, all this, some blueberry. Yeah, we talk about a lot of blueberry nutritional stuff. So maybe also in the future we can do some project with him too. But either way, what we have been doing, what Dr. Li and also our team, all the professors in Nanjing Agriculture University, we are doing the same way. So we are on the front line of the science. We are not aiming behind of the current technology of the fertilizer. And we talked to some actually fertilizer company. This is a company, I think that company was, some people are Spanish people. In Florida, it's a lot of Spanish people. You don't have to speak English as long as you can speak Spanish in Florida, because they have a lot of, most maybe half more people are coming from Latin America, so they all speak Spanish. And the guy in the white color, he speaks perfect English. And me, I'm not speaking perfect English. I can speak English, but not perfect. And on the right side, he's my level. We understand each other, but it's not a perfect English. But what they are doing the same thing like we do. They have all different fertilizers. We are doing in the same. So we keep the exchange. We acknowledge, each other. Our science, our fertilizer are the best in the world. So when you go to the international fertilizer shop, basically you see the newest technology. What is the best fertilizer on the market or in the development? So we see that, and we compare our stuff. Our greenwood fertilizer are the best fertilizer in the market. This, I didn't get this from the fertilizer show. This is from the other analysis. So this, you can see, today is 2025. This graph is to 2022. Why you need to go to the conference? Why you need to go to the show? You can get your science knowledge from reading, from the library, from the publication, from the journals. from the articles. But one thing you can see from this chart, when you get all the information from the computer, from the library, from the newspaper, all this published data is about 2-3 years difference. When you see the data, when you see the paper was finished like 2-3 years ago, the data. So you don't see the current technology in the paper. When you, if you want to copy anything from the book, from the book is even worse, maybe 10 or 15 years or 20 years. When you see, oh, this method is nice in the book, I copy it, now you're too late. That book, writing book, take many years. I published my book in 88, I don't remember. The book, published book is it take many generations, many years to write, and when you publish it, it's a very delayed technology. When you find a paper, research paper in the journal, on the newspaper, you even publish it today, the data it was 2 years ago. Because when you write in paper, the paper you finish writing, you give to the journal take a year to review, to say, okay, your paper is passed, we can publish it. And take another year to publish it. So from your writing to publish it, it takes 2 years. From your writing, when you finish your writing, that writing data is 3-4 years ago's data. So totally like 6-7 years. When you see the paper, the data is 6-7 years old. Like we see the sunlight. We see the sunlight. We see the sun, the moon, the star. When we see the star, it's not the star they are right now. The star is maybe 100 years ago. Because the light travels for a long time to your eye. So you see this one of the moon or something as a Mars. When you see the light coming, it is not the current light. It is many years ago's light. You see a history of the light. That star maybe already disappeared when you see the light, but you see, oh, that's a new light now. The same thing applies to scientific paper. When you see the paper, that the data is already 6 years old. So that's why we need to go to the convention. We need to go to the show. We need to go to the meetings to meet the people to see what are they actually doing this work. What are they doing now? The data they are collecting right now will not be seen in journal, in paper, in another 3-5 years. So that's the reason we need to go to the convention. Normally I go to the agriculture annual convention every year. So they will keep updated with all the knowledge and keep communication with all the people, all the professors you work with. So what is the future of the fertilizer? That's what we discussed a lot during the meeting. Conventional fertilizer, NPK, is still using. Most fertilizers are NPK, granule, liquid, all kind of fertilizers. Still using, but it's not the future because it's a chemical, because this is not organic. So the future of the fertilizer is organic fertilizer. Not only our fertilizer is from plants and animal materials by digesting, by enzymatic, by digestion to break down the plant and animal material to become small molecules, become water soluble, and the plants can, all the nutrients, amino acids plants can use. So this is totally organic. Organic fertilizer is the future. And also, another future of the fertilizer is we need to use some microorganisms, like bacteria. The bacteria, some legumes, some like soybeans, the roots, they have the bacteria, they are symbolic. They live in the soybean roots or faba bean roots. When you pull out the soybean plants, you will see little bit like beads like on the roots. They are a ball, a beads inside are bacteria inside. The bacteria inside the roots. So plants needed the bacteria. The bacteria needed the plants. And they help each other. These bacteria can fix nitrogen. The air we breathe, the air think it have above 70% nitrogen in the air. So much nitrogen in the air. Why plants cannot use this nitrogen? We don't need this fertilizer anymore because the air have all this nitrogen in the air. The sad thing is plants cannot use them. The plants cannot use the nitrogen in the air. They need bacteria to gather this nitrogen from air to fix into the soil. So that's why when we do the planting, the agriculture, we do change crop every year to called alternation. You can this year use a wheat, this year plant a rice, this year plant corn, next year you plant soybeans. Because the corn uses a lot of nutrients from the soil. And the next year you plant soybeans. So soybeans help to add some nutrients into the soil. This way you are not only use the soil, you actually to enrich the soil every year, so make your soil long-term sustainable agriculture. So we can use this kind of bacteria put into the fertilizer, or we just spray this bacteria into the plants. And also like the bacillus, this genus of the bacteria. When you spray this bacteria into the soil, into the plants, they can make a lot of nutrients available. Sometimes you test your soil. Your soil is not a lack of nitrogen. You have enough NPK in the soil, but the plant just cannot use it. Why? Because this NPK, this a lot of nutrients are not, they are in the soil like nitrogen in the air. They just not water soluble, not available for plants use. Plants are very picky. They only pick when the nutrients in the movable, in the water soluble stage, and they can move the nutrients with water to get into the root system or from the leaf then you can help the plants. Plants can use these nutrients to help them photosynthesis or synthesize other nutrients, other proteins or carbohydrates. So these bacteria not only fix nitrogen, they also help facilitate the nutrients metabolism, make these nutrients available for plants. The idea is very nice. The method is not easy. I think it's at least 100 years work have been done. Not much have been achieved. Dr. Li's professor, Dr. Fan, he was the father of microbiology in China. He was also senior professor when he was in US to study this nitrogen fixing bacteria. Lifetime, several generations, not easy. So it depends on the new technology, how to culture this bacteria. When you microorganisms, it's not easy. I have a master's degree of the microbiology, molecular biology. I know the trick. You see, it's easy to culture a bacteria. Very, very difficult. My PhD program is about a bacteria. It's not easy. Some people before me, they spent over 10 years. They never got the bacteria species I got. But I got it right away. In a month, I got the bacteria. Some people, many of my coworkers in my lab, they spent over 10 years, they never got it. So when I started, I got it. Is it lucky? Not necessarily. It's also a lot of your observation, your knowledge base help you improve your methods to culture. Not only sterilization, your autoclave help you minimize all the contamination. You have all other stuff, so much, the nutrients of the media, everything, the temperature. You check with the plates every day or you check your plates every week. Make a lot of difference. So the culture of the bacteria, the isolation, the selection of the bacteria is many species. You don't know which one is good. You have to do a lot of selection. Maybe you need to do some gene modifications. Change some bacteria. Maybe it is ideal for the nitrogen fixing. Also, it'll be easy when you have nice workable bacteria. You know it's help, it works. And how do you put this bacteria into your plants? You spray on the plants. It's not like bacteria work on the human. We just cut it, our hands open, with skin open, and put some dirt on it. We got infection. No, for plants, fungi, it's easy to get into the bacteria for the plants. The bacteria is not easy to get into the plants. And get into the plants, they have to, for the pathogenic bacteria, like a diseases, like spots, leaf spots. These kind of bacteria can easily get to the plants. For the symbiotic bacteria, the nitrogen fixing bacteria, it's very, very difficult because we need to inoculate the bacteria to the plants, and the plants, they need to like it. When you disease the bacteria get in the plants don't like it. They eject it. They create something. They make a burning. They just leave the cell died. When the cell died, and the bacteria died out. So that's why you see the leaf spots. When the leaf spots, because a bacteria put on get into the leaf, and the plants, they initiate the defensive system mechanisms. They make a circle around the bacteria. All the circle along the circle, all the plant tissue, they just die off. Okay, you want to attack me, I'm going to die. And then what do you do? The bacteria die too. So this is not a symbiotic. This is diseases, the infectious kind of parasites. The plants host don't like infectious pathogenic bacteria. They kick it out by sacrifice, by dying a line of the tissue cells, and they make a hole, the bacteria lost. For the symbiotic bacteria, the nitrogen fixing bacteria, we need to inoculate it. And the plants say, oh, you are aliens. You are not a part of my plants, but I like you. Okay, I make a chamber for you. My leaf, my roots make it a little bit swollen. Bacteria come in, stay in my roots, and the bacteria find the home, find the host loam. Very nice. I also give them nutrients. And the bacteria, okay, you give me a very nice water and all kinds of carbohydrates, sugar, everything. Okay, I'm going to get some nitrogen for you. And they get some nitrogen from air, fixing to the roots. So bacteria help the plants. Plants host the bacteria. They are symbiotic. They together. Very difficult, very hard to find a species of bacteria, and that plant species can take, and they can coexist for a long time or for the future, many years, generations. Very, very hard. That's called the future of biofertilizer. You don't know what happens when.